Just finished all of anything related to neurophysiology. So we're finally done with that bit. And the reason I start with that is because central nervous system, peripheral nervous system, that is our direct communicatory pathway to whatever we're looking at, whether we're talking about the skeletal muscle, glands, any of the tissues that we're going to be looking at. Now we're going to be specifically working with the endocrine system, which is going to use a different type of communication tool, which is going to be our chemical hormone. So what we need to do in our endocrine system, we'll just do a quick overview of what we're going to be talking about. So what we're going to do is we're going to do a quick overview of our endocrine system, the endocrine system a little bit. We're going to talk about homeostasis and our feedback loops, just things that we've already seen before. Then we're going to talk about the purpose of the endocrine system to communicate. So we're going to talk about the central endocrine organs as well as the peripheral endocrine organs that for the sole purpose of sending communicatory molecules out through the circulatory system. And so what we're first going to take a look at, we're going to take a look at the hormones. We're going to start off with just the basis of how we classify these hormones versus based on their chemical makeup. We'll take a look at how cell signaling works, as these hormones work for cell signaling. We'll take a look at the effects of them, and then how we regulate the amount of hormones in our systems. And then we're going to branch off into the endocrine system. We'll start taking a look at how we produce these hormones, which organs are going to be responsible for the production of hormones, and we'll talk about those pathways to support those same functions that we're going to be able to see that for all the pathways that are related to the endocrine system. So we'll first start off with our central endocrine organs, and then we'll talk about our peripheral endocrine organs on Thursday. So first thing we're going to start off with is to just revisit negative feedback and positive feedback, which you guys should be well versed on the differences between the two, because we've kind of beaten this over and over and over again. Negative feedback is something that is going to resist a change. So if we see an increase in blood sugar, what we're going to see it as the efferent pathway is a reduction in sugar. So increase in sugar will result in a decrease in sugar. And this is where we saw our body's ability to maintain homeostatic values. We had a narrow range of what we're looking at. We want to make sure we can maintain that, whether we're looking at sugar, we're looking at body temperature, if we're looking at amounts of hormone in the blood, etc. Now, positive feedback is something that is going to strengthen the change. This is our reinforcement. And here we're going to be able to explore the one hormone that we'll talk about that we've talked a little bit about is oxytocin. And the stretching of the cervix is going to send information to the control center, which is going to result in oxytocin, which will amplify, so an increase. So this what starts off with the stretching of the cervix will lead in an amplification of that signal. So that oxytocin is going to be the hormone that's involved that will cause uterine contraction. This is the efferent pathway. And that uterine contraction will stretch the cervix even more, which will result in more oxytocin. So if you were to measure the amounts of oxytocin levels, especially towards birth, you would see a huge rise. And that's why we call it positive feedback. And all of these responses here in both pathways, you're going to see similarity in the setup, in which you're going to have our sensory. These are the receptors that are going to send information over to the control center, which we now know is going to be our central nervous system. And the effector pathway is going to be the motor pathway. And motor, maybe not necessarily in terms of motion, but motor as the efferent pathway. This is our motor response. And what we'll note is the sensory information that leads into control center, that setup is basically the same. But The main difference between negative feedback and positive feedback is the efferent pathway. So efferent pathway in the negative feedback will reverse the change, while the efferent pathway in the positive feedback will reinforce that change. Next, what we're going to look at is how we derive our endocrine glands. And so what we're going to be able to see are the surface epithelial cells, and you'll see an invagination of that surface epithelium. And towards the very bottom of that surface epithelium, you're going to see cell specialization, differentiation of that cell to become cells of a secretory nature. That basically means that they will start to produce something. While these epithelial cells are meant to protect, these cells will differentiate to become cells that will release a product. And so our exocrine glands are the ones that are going to produce things that will get secreted out into the lumen. But what we're really going to be talking about in this lecture is specifically taking those secretory cells and breaking down the connecting epithelial cells. So we're going to have this pocket of secretory cells that will function on their own. And we're going to have blood vessels that are going to wrap around that endocrine tissue so that we can, anything that's secreted will make its way into the circulatory system. So that's how we're going to be able to take a look at the endocrine organs and how they produce things that will have a systemic effect, meaning it's going to affect anything that circulatory system can deliver to. Looking at our solutions here, this is going to help describe our tonicity. How do we maintain proper levels of water in our system? We always want to maintain an isotonic solution. And remember, tonicity refers to the amount of solute in our solvent. Okay. So specifically, this will be stuff on the outside. How concentrated is our solution that our cell is in? And so an isotonic solution, we would know that we have net movement of water equal to 0. Meaning we have the same amount of water going in and the same amount of water going out. So osmosis in this point is pretty much canceled out. We got equal movement on both sides. Now hypotonic solution means low solute in our aqueous environment. Hypertonic means we have a high amount of solute in our concentration, in our solvent. And so these are going to have a profound influence on how the cells are going to function due to the movement of water, or osmosis. So as I mentioned in our YouTube example before, if we have a lot of solute on one side, water is going to want to move over to the right side. And remember that example that I used before. Water is going to want to move in the direction to balance out the concentrations. So in this case, in our hypotonic solution, we have an increased net movement of water into the cell, because this is where our solutes are. We have less solutes on the outside, so therefore water is going to move over into the cell. And that will end up inflating our cell, eventually to the point that it can burst, which we're going to be able to see called hemolysis for our red blood cells. Hypertonic solutions are the exact opposite, where water wants to move out into an area with hypertonic solution. We have water wanting to move into areas where there's more solute. Therefore, we would see cells that undergo crenation or shrinkage. So you can see from here that it's important to maintain that balance in order to ensure proper functioning of cells. In this case, our red blood cells, we want to make sure that water movement in and out is balanced. We'll start to take a look at cases where we have water imbalances. What happens when we cannot regulate the tonicity of the fluids moving through our body. And the last bit here is to take a look at our bottom right. Here we can see our epithelial cells. We have our tight junctions holding them together so that things can't move through in between the cells. And these epithelial cells are meant to create that barrier from the lumen to the extracellular fluid. So all this right here comprises the extracellular fluid, which is going to be a combination of the blood, the capillaries that are going to allow fluid to flow through the circulatory system, as well as the surrounding interstitial fluid. That's the fluid that these cells are basically bathing in. So we're going to be able to see the exchange, especially when we get into our circulatory lecture. We'll see how fluids will move from the blood to the interstitial fluid, and from the interstitial fluid into the cell. It can't move through the tight junctions because that's where we're blocked off. So things have to move through the lumen, through the epithelial cells into the lumen, or from the lumen through the epithelial cell back into our circulatory system. This is going to be our exchange. How do we keep things out in the lumen, and how do we get things to move through? And that we'll talk about more in our digestive lecture. And then just to quickly revisit our biochemistry lecture, we'll take a look at lipid-soluble molecules. So lipid-soluble molecules are typically things that have high hydrocarbons. We'll see some of these complex structures, but they typically will exhibit covalent bonds, so nonpolar covalent bonds. So these nonpolar covalent bonds means that these electrons that are floating around spend equal amount of time around the molecule itself. Now, the thing is about lipid-soluble molecules is that they can typically move through the cell membrane. So if I were to draw a quick cell membrane, here's our phospholipid membrane. Lipid-soluble molecules can typically flow through. Now, the other hand, water-soluble molecules are going to be polar. So they will not be able to move through. Typically, water-soluble molecules, due to their high polarity, will not be able to pass. So the effect of these hormones that we're going to be talking about, we're going to be able to differentiate between lipid-soluble and water-soluble. How are we going to get the effect on the inside of the cell, the important part of the cell itself? How do we get this cell to exhibit some form of change when these types of hormones come into play? Now that, as I mentioned earlier, our hormones are going to be flowing through the circulatory system. So we have to wonder, how are we going to have lipid-soluble? Again, these are things like fats. How do we get fats to dissolve in water, specifically blood plasma? We'll learn about that in just a little bit. Water-soluble molecules, they can pretty much dissolve in blood plasma on their own. So we'll, again, explore the importance of all of that. Now, glucose metabolism, again, these are the things that we talked about in the tail end of our cellular respiration lecture. I had you guys go in and understand the different terms, glycogenesis, gluconeogenesis, and glycogenolysis. So that you should be well aware of. Now, the purpose of glycogenesis is to store excess glucose. In times when we have excess glucose, we want to be able to store that so we can use it later. So this is where we see glycogenesis taking place in the muscle as well as the liver itself. And the term here you see hyperglycemia is excess glucose. Now, glucose release, these are things that we typically see with this as a sympathetic response. If this is a parasympathetic response, right, rest and digest, as well as the storage of glucose, this would be predominantly a sympathetic response. This is our way of releasing glucose. And we can release that from the liver, and we can release it from the muscle. Now, we can't burn glycogen in cellular respiration. So what we're doing is we're taking that glycogen and lysing it to increase the availability of glucose. And that glucose can then undergo cellular respiration through glycolysis, through the Krebs cycle, and then through the electron transport chain to generate ATP. So this whole process is to increase the amount of glucose levels in order to generate that 36 ATP per glucose molecule. But glycogenesis is the generation of glycogen. And gluconeogenesis is turning lactate into glucose in the liver, which as we saw. And glycogenolysis is the breakdown of glycogen to glucose. Now the hypothalamus we'll revisit. This is going to be the first of our central endocrine organs. And again, something that we'll talk about a little bit later on. But this is our endocrine organ in the central nervous system. So we'll start with that. That is the major endocrine organ that is going to be able to regulate the numerous activities of the human body. Things like hunger, sleep, wakefulness, emotions, sexual arousal, anger, fear, pain, pleasure, all of that. We'll talk about those activities. And we're going to be able to see how, when we visit at the very end of this lecture, we'll see how the hypothalamus can communicate over to the pituitary, which is going to be the second in line. So typically what we're going to be looking at, we'll be looking at the hypothalamus, which will send hormones over to the pituitary. And that pituitary will send out the hormones to the peripheral endocrine organs. And you'll see that some of the hormones that we'll be talking about in the pituitary are going to have a direct effect on some of the tissues. And we'll visit those in just a little bit. So what we're talking about today is the hypothalamus, pituitary to the direct tissues, and then we'll look at the other peripheral endocrine organs on Thursday. And what we also want to take a look at through this entire process is how negative feedback and positive feedback come to play. Because as we get these hormones that are produced down the line, they'll come back and have a direct effect on the hypothalamus. And that will form a process for negative feedback. So what starts off with the production of a hormone, such as testosterone, will start from the hypothalamus to the pituitary, over to the gonads, or to the testes specifically. And that testosterone will go back to inhibit that production. When we looked at our nervous pathways, those nervous pathways, we talked about the direct excitation or inhibition of the target tissues.
I have a question.
Yes?
So when the signals are sent from the tissue to the pituitary gland, those are, tissue to pituitary gland is a negative feedback?
These are just going to basically employ some form of negative feedback. So I don't want you guys to get too lost right now about the pathway. That's what we're actually going to go into detail. So all I'm trying to get at with my descriptions here is just pointing out that there is a form to turn off the production. What starts off with the production of that hormone is going to result in the turning off of that production. Because we don't want too much of that hormone. We only need a certain amount to trigger that systemic effect throughout the whole body. To give you a good example would be puberty and what's going to result in bone growth. When we start producing that hormone, we want to see that hormone come in, have an effect on the body, and some form to turn that back off. Because we don't want too much of it, we only want just a little bit of it. So, does that answer your question? So looking at this, we'll see, as I mentioned, the nervous system is going to be a fast connection. Whenever we're looking at the skeletal muscle, when we're looking at the smooth muscle, or we're looking at any of the glands, that was a direct, fast pathway, an electrical chemical pathway. We saw action potentials traveling down an axon. We saw neurotransmitters diffusing across the synaptic clefts and targeting the tissues. So that's a direct target. So if I'm to take a look at a muscle and how it gets to contract, we saw a motor neuron that would target that muscle directly. Now the endocrine system is going to be specifically a method in which we're going to be dumping these hormones into the circulatory system. And in order to get that response, that target tissue has to have receptors that receive that hormone. So in order for us to get into that, we do have to talk about how that hormone, the mechanics of that hormone to flow through the circulatory system and how it's going to bind to that tissue specifically. And so when we see how this pathway evolves, whether we're going to see the pituitaries releasing that hormones, it's going to have a very slow mechanism and a slow but prolonged effect. To give you an example, we talked about the epinephrine that's released by our postganglion neurons in the sympathetic pathway. We saw epinephrine and norepinephrine. Whatever epinephrine as a neurotransmitter would target that target tissue and cause direct excitation. Now we see epinephrine as a hormone as well, and that we'll see in just a little bit. is the hormone that's released by the adrenals. And that hormone makes its way through the circulatory system and creates a prolonged effect. So if you think about that fight or flight, it's not a very fast excitation. Well, it is a fast excitation, which is a result of the nervous pathways. But the prolonged effect of the sympathetic pathway, the prolonged effect of that sympathetic response, elevated heart rate, elevated respiratory rate, lasts quite some time. And it takes some time for you to kind of calm down from that sympathetic response. That's due to that circulating epinephrine through our system. And eventually, parasympathetic pathways will come in and take over. And eventually, that hormone will get broken down. So let's take a look at the types of hormones. So now that we've described hormones as a communicatory molecule, we can see how they are going to have a direct effect on that tissue. So every hormone that is released by the endocrine tissues will have to target a cell. And the way it targets that cell is through a receptor. So we have a hormone that binds over to a receptor on a cell. And so that effect of those hormones can have a local effect, meaning the hormone only affects the cells close to where that hormone is released from, or it can have a very long effect, meaning it can travel throughout the entire body to make it to its target tissue. So the ones I'm describing are local versus distant. To give you an example of our local, let's first break down the two types of local. Local hormones, we have autocrine and we have paracrine hormones. So autocrine hormones are the types in which you have the production of hormones that come in and target the receptors on the same cell that released those hormones. Now, paracrine hormones are the ones in which cells release hormones that target nearby cells. What examples would we see of paracrine hormones? That I can give you an example would be like a histamine response. If you've ever gotten bitten by a mosquito bite, what happens is it's going to create histamines that are released by cells that will come in and cause an inflammatory response to all the cells nearby. So that's where you get that exaggerated effect, and it becomes quite raised and inflamed. Now, distant hormones are ones that are going to get sent down the circulatory system. So here you can see an endocrine cell releasing those hormones, and those hormones are going to end up circulating through the circulatory system. And wherever those hormones have their effect, they'll get released out, and they'll target any cells anywhere down the line that are going to have receptors for that hormone. So you can see how that can take effect. So if you were to think about hormones that are released from the pituitary, they can make their way over to the reproductive organs. They just have to circulate through that circulatory pathways to those target tissues that have those receptors.
What would be an example of an autocrine?
So here we divided where the effect of the hormone is, whether it's on the same cell, nearby cells, or distant cells. Here we can divide the classes of hormones based on their molecular structure. So we have two different types, again, like I mentioned earlier, lipid-soluble and water-soluble hormones. So lipid-soluble hormones, given their properties, and we'll see this in a couple slides, bless you.
Thank you.
We know lipid-soluble hormones can diffuse across the cell membrane. So if I have a cell membrane on here, I know our lipid-soluble hormones can travel directly into the cell, while the water-soluble hormones can come in. They won't be able to travel through the cell, so they're going to involve some form of receptor. And that receptor is going to have some sort of target that's going to create a response on the inside. So we're going to involve what we're going to refer to as our secondary intracellular messenger. So let's take a look at our lipid-soluble hormones. The first of these, of which you can see are lipid-soluble hormones, will have testosterone, aldosterone, and cortisol, which are all steroids. And how do we characteristically define steroids? We can see that we have 4 ringed molecules. And each of these differ. They all have the same structure, in which you're going to have 3 6 carbon rings with a 5 carbon ring hydrocarbon. And all of them, for the most part, have the same base structure, but they do differ on the attachments, the functional groups on each of these. So while they may be similar in molecular composition, they do vary based on their properties and what they target. So given that these are lipid-soluble hormones, they're not proteins. They are, for the most part, lipids. And so they are going to be made by smooth endoplasmic reticulum. So the smooth endoplasmic reticulum will then package these up by the Golgi apparatus, and that will get shipped out. So here you can see testosterone. The function is testosterone, which we'll be able to see, aldosterone, and then cortisol. Cortisol, very similar to corticosteroids. Things that are going to be produced by the adrenals, which we'll see later on. Now the next type of lipid-soluble hormones is what we call our thyroid hormones. And thyroid hormones we'll be able to see toward the bottom. It's going to consist of T3 and T4, which we'll describe later on. But T3 is also known as triiodothyronine, which triiodo refers to 3 iodines attached to a modified thyrine molecule, or tyrosine molecule. And here we can see this large molecule with 1, 2, and 3 iodines attached. So we'll see how that's going to have an effect on metabolism. Now the next one is nitric oxide, NO. And nitric oxide is something, even though it is not polar, you're going to see how nitric oxide is going to have an effect, something that we typically see in autocrine as well as paracrine signaling. So nitric oxide is just one of those things, I actually just read today, there was, I don't know when it was discovered, but it was actually discovered at UCLA. And its profound response and I'll show you that on our next slide. Nitric oxide, it has a response in endothelial cells in the blood vessels. So it is a potent vasodilator. And you'll be able to see this in our circulatory lecture when we talk specifically about the cardiovascular system. So this nitric oxide, not to be confused for nitrous oxide, which is laughing gas, nitric oxide is something that is actually produced by endothelial cells. And they do this in order to regulate the amount of vasodilation that occurs. So if I can just quickly draw a blood vessel, we have smooth muscles that wrap around those vessels. And nitric oxide can be released in, by these smooth muscles that will end up causing those smooth muscle cells to relax. So this has a very profound effect. If you were to take a look at some of the clinical things that they can typically inject just a little bit, just measure the amount of nitric oxide that's given off. And that will essentially regulate how much vasodilation, how much do we dilate those vessels. So looking at the other steroid hormones, you'll see the steroid hormones and where they're going to be secreted, and then we'll talk about the effects of those. And the thyroid hormones, as naturally secreted by the thyroid gland, and how they're going to have an effect on metabolism. And you'll see the main difference between T3 and T4. We'll talk about that when we get to our thyroid lecture. All right, so now that we have our lipid-soluble hormones flowing through the circulatory system, let's see how they exert their effect on the cell itself. And I do want to skip ahead a little bit and talk about what the main effect of these hormones are. So you'll see a little bit of this. I know I'll go into a little bit more detail about it. But basically what's going to be taking place, and I'm not really talking too much about these, I just wanted to point these things out. Endocrine hormones are going to play some physiological role on their target cells, depending on what those cells are. There's a whole bunch of them that they'll take place. And that is all going to involve some way of communicating, this one I want to talk about, some way of communicating with the cell to do something. So whether it's a growth hormone type cell or it's testosterone, you're going to want that cell to start producing a bunch of stuff. Okay? Okay? So you want some effect to take place in the cell. So the question is, how do we get that hormone to communicate with the cell itself? How do we exert some sort of change in the cell in response to that hormone binding to that cell itself? So let's go back to our lipid-soluble hormone. So we're talking specifically about our insoluble in blood plasma hormones. So here we would see a free hormone. If you take a bunch of these lipid-soluble hormones and you put them in blood plasma, you have oil and water. These two don't mix, right? Right? No, they don't mix. So how do we get these lipid-soluble hormones, these fats and steroids and cholesterols to dissolve into blood? We utilize a transport protein, and the transport protein in this case is albumin. If you ever take a look at blood labs, what they're looking for albumin. And albumin is a carrier for it. So if you notice that someone has some deficiency in albumin, well, they most likely will have some heavy hormone imbalance because these lipid-soluble hormones, testosterone, estrogens, calcitriol, and aldosterone, all of those really can't be dissolved in blood. So 90% of all lipid-soluble hormones will be dissolved via albumin, and it acts like a carrier. The same way soaps exert their effect on lipids on our hands. So if you're washing your hands, most of the time you want to use soap because all the fats that are on your skin, if you try and wash away with water, those lipids don't dissolve into the water. You utilize soap, which comes in and surrounds and carries those lipids. It makes those lipids soluble, and that's exactly what albumin does. It works in that same mechanism. So this, if you see free hormone, you have about 10% that will be dissolvable in blood, and the other 90% are carried by albumin. Now that we have that lipid-soluble hormone flowing through the circulatory system, those free hormones will end up diffusing out into the interstitial fluid surrounding the target cell itself. So that way, as these free hormones dissolve, you'll get these transport proteins that will help unload those free hormones back into the circulatory system as it travels through. Now we have these lipid-soluble hormones, and the first thing I mentioned about them is that they can travel through the cell membrane. So they diffuse right into the cell itself. So what is that hormone going to do now that it travels through the cell membrane? And all of this is described for you in the next slide over, so you don't have to furiously write this stuff down. I've got it all broken down for you. The next thing it's going to do...
I have a question.
Yes?
But the albumin, does it carry 90% of the hormones, or does it dissolve that? Does it carry or dissolve?
So what it does is it basically locks itself in. The protein comes in and it has a binding area for these hormones. It'll allow it to basically help dissolve into the blood. It doesn't really do anything for the hormone action other than to just carry it in the circulatory system.
And the other 10% doesn't dissolve?
The other 10% is just dissolved freely in the blood itself. Even though I mentioned it's not completely soluble, some of it will dissolve, but it's not enough. And we need more of it, and that's where albumin comes in. So now that you see the hormone making its way into the cytoplasm, where is it going to exert its effect on? Well, the receptors for that hormone. Remember, that hormone has to have the receptor that receives that hormone. That hormone makes its way through the nuclear membrane and binds into a receptor. Okay? And this receptor has two active sites. It has one specifically for the hormone, and it has a site that binds to a specific area on the DNA. Okay? So that area on the DNA is called the response element. Okay? So there's a certain area that responds to it. It's a specific targeted area, the hormone response element. So here you get the receptor and hormone complex that comes in. Once that hormone binds to the receptor, now that receptor hormone complex can come in and bind to that DNA. So once it binds to that DNA, it's going to activate that specific area, that gene specifically. Because as I mentioned, that cell needs to do something. It needs to produce a bunch of something. So now that it binds to the area, we can undergo transcription, where we transcribe that DNA into RNA. And if you remember from our earlier cellular lecture, We talked about transcription, we talked about the production of RNA, we had our SNRPs that come in and excise all the exons and use that mRNA. And that mRNA, as you saw in our labs, will serve as a template for the proteins that will be formed. Okay. So once we have that mRNA will make its way out of the nuclear complex and will bind to a ribosome, and it'll undergo translation where we get the production of our proteins. So here you can see the direct effect. You see the effect of that hormone make its way directly onto the DNA itself. Granted, it still uses that receptor, but it has that direct effect into it, which results in the production of that protein. So the whole purpose of this is what we call, that you'll see in a later slide, called upregulation. Upregulation meaning that we're finding that specific gene that hormone targets to result in the production of a bunch of those proteins. Because what could those proteins do? They can differentiate and turn into enzymes. They can differentiate and they can become channels. Any way you look at, we have excess production of those proteins, whatever they may be. So this is what we call upregulation of a gene. So if you were to think about one of those hormones that I mentioned, testosterone. Testosterone that can make its way into can result in the production of skeletal muscle. So these would come in. If you think about testosterone making its way into a muscle cell, testosterone comes in, binds to that hormone, that receptor, which then binds onto the DNA to start transcribing the mRNA that encodes for muscle proteins. And those muscle proteins will end up getting developed and so forth. So here we see increased activity. And we know mRNA doesn't just create one protein, we can create multiple proteins. So here we see a slight amplification of the signal. We get one hormone that binds in, which can create a bunch of mRNAs. Those mRNAs can come in and then create more proteins. So all of that is described for you on here. So all of this is a direct effect. That lipid has a direct effect on transcription of that gene.
Is it upregulation with turning into a channel or enzyme?
Not necessarily. Upregulation is activating of a gene. So if you activate that gene, you can start producing the proteins that gene encodes for. Now let's take a look at our water-soluble hormones. And our water-soluble hormones, obviously, are all water-soluble because they have polar covalent bonds. Specifically, the molecule exhibits strong polarity in that water, that hormone can dissolve in water. And we see the main type of these types of hormones. If we saw our lipid-soluble hormones being lipids, we will see that our water-soluble hormones are all types of proteins, whether they're small peptides, longer proteins, or just the amino acids themselves. And a good example of all of these, we'll take a look at serotonin. We'll look at dopamine. I know these are also neurotransmitters that you saw, but they also function as hormones. Epinephrine. Another one I want to talk about is insulin. Insulin is a big one. Histamine as well. Melatonin we'll see later on. These are all water-soluble hormones. And again, protein hormones have multiple polypeptide chains, meaning they've got a bunch of peptides that are put together in quaternary structure. And a good example of these are insulin and glucagon. Our peptide hormones are just amino acids that are strung together, of which we have growth hormone and oxytocin. And then amine hormones are basically modified amino acids, of which we have epinephrine, serotonin, dopamine. Again, things that you saw were similar in construct to our neurotransmitters. The last of these branches are called eicosanoids. And these eicosanoids are derived from arachidonic acid. You don't need to know that. But the structure of these arachidonic acids is that they're amphipathic, meaning they have both polar and nonpolar. So they are kind of like phospholipids. And the two that we'll see on here are prostaglandins and leukotrienes, which we'll describe on the next slide over. Now, looking at these on here, you'll see the water-soluble hormones and where they're secreted from. And again, most of these we'll talk about where they derive from. We won't really be talking about serotonin. I won't really be talking too much about histamine, but we will be talking about epinephrine, norepinephrine, melatonin. We'll be talking about all the hypothalamic releasing and inhibiting hormones, oxytocin, antidiuretic hormone. All of these we'll be talking about. Things that we'll be talking about in our peripheral as well as our central endocrine organs. The last two on here, again, are icosanoids, are prostaglandins and leukotrienes. And these prostaglandins, you'll note, are produced by all cells besides the red blood cells. All of these hormones are produced by by cells that have endocrine functions. And prostaglandins and leukotrienes, prostaglandins specifically for pain, and leukotrienes specifically for inflammation, are actually newer. They are newer hormones that have been studied simply because they are produced by cells that are not necessarily endocrine in nature. So we produce quite a bit of them. So prostaglandins are things that are released as a result of pain, and leukotrienes as a result of inflammation, things that have a direct effect on autocrine and paracrine in nature. I don't really go into too much detail about it, but really the main idea I want to get at is the mechanism of water-soluble hormones. So let's take a look at how that works on the next slide over. So water-soluble hormones are water-soluble and therefore dissolve in blood readily. So they don't need a carrier. So they'll freely be dissolved into the blood, and again, they'll diffuse out into the interstitial fluid surrounding the cell. And again, all the notes that you're going to be able to see are on the next slide over. So looking at this, we'll have that water-soluble hormone that comes in and cannot cross the cell membrane. So therefore, where do we expect to find the receptor? We expect to find the receptor specifically on the cell membrane itself. So that comes in and binds directly to the receptor, and that's where that hormone stops. It doesn't cross through the cell membrane. Therefore, we need some form of signaling to take place inside the cell. So how do we do that? That receptor comes in and targets this G protein. And G protein is like a molecular switch. Once activated by the receptor, comes in and swings over to the next molecule right next to it called adenylate cyclase. So as that G protein comes in, activates adenylate cyclase, this is where we're going to be able to see ATP getting converted over to what's called cyclic AMP. And cyclic AMP is the second messenger. So if our outside extracellular messenger is the water-soluble hormone, the intracellular messenger is this cyclic AMP. So we see the cyclic AMP, and that is going to be the thing that comes in and creates this whole protein kinase cascading. And I call it cascading because what it can do is it comes in and turns on proteins, or comes in and turns on enzymes. So we activate an enzyme that can activate another enzyme that can activate another enzyme. Down the line, it will end up turning something on. So if you ever see the term cascading or cell signaling cascading, that's what it's referring to. It's this whole turning on something that turns on something else that turns on another thing. And it doesn't necessarily have to be that complicated, but that activated enzyme comes in and can turn on a protein. So that protein comes in, and we can phosphorylate that protein via that same enzyme that was turned on. So what we can see, an example that I can give you of this, is epinephrine. As I mentioned earlier, epinephrine is one of the water-soluble hormones that is secreted by the adrenal medulla. And what that epinephrine is going to do is it's going to come in and target and bind to that receptor, which results in that cyclic AMP. That cyclic AMP can come in and now increase the amount of glucose availability. So what it does is it can turn on the enzymes that can turn glycogen into glucose. So activate those enzymes, now you have glucose that's readily available, and now that'll come in and start generating ATP. Again, part of the sympathetic response is that quiz question that I gave you. So epinephrine, as part of the sympathetic response, can result in the increase of glucose availability through glycogenolysis. In this case, we do mention that this is an indirect pathway. Why is it indirect? Because that hormone doesn't directly affect the production of cyclic AMP, or doesn't come in and affect the protein directly. We have to utilize a second messenger, which we know is cyclic AMP. We have a huge amplification of the response, as you can read on the next slide over. That hormone that binds to the receptor can actually come in and turn on multiple G proteins. So imagine that one epinephrine molecule coming in, binding to a receptor. That receptor turns on 3 G proteins. Those 3 G proteins come in and activate 6 adenylate cyclases. Those adenylate cyclases then produce 1,000 cyclic AMPs apiece. Okay? Now those cyclic AMPs can come in and turn on a protein, which can turn on multiples, which can turn on more and more and more, all through the binding of that one hormone. How do we turn off cyclic AMP? Phosphodiesterase, again, an enzyme that comes in and breaks down cyclic AMP to turn off that internal self-signaling. So this is a huge amplification of the response, or in a way, is a form of positive feedback.
The amplification is the positive feedback?
Yep. So we see, think of that stampede, that cow stampede that I mentioned earlier. It starts off with one cow freaking out, results in an amplification of that response. We have multiples coming in to turn on multiple enzymes. Now all of this looking at our hormones. Now that we've been able to describe our lipid-soluble and our water-soluble hormones, we have to mention the receptors, because that's always something that we've ever looked at. If you ever look at the history of studying of hormones, they've only looked initially looking at the hormones themselves, but they also have to study the receptors. When you ever study diabetes mellitus, the issue is not necessarily, and this is specifically type 2. I want to talk about type 2. Type 2 is what we call insulin resistance. It's not an issue with the insulin or the production of insulin per se, which is type 1 diabetes. Type 1 is an autocrine disorder, an autoimmune disorder where our own immune system comes in and targets the pancreatic cells that produce insulin. Therefore, people that have type 1 diabetes typically get them at a very early age, and they have to have a pump that puts insulin into their bodies. Now, type 2 diabetes is what we call insulin resistance. And insulin resistance is where and we'll be able to see this in a later slide, this is where insulin comes in and has to bind to the receptor. So the issue is not insulin, it's actually the receptor itself. So why do I mention this? Is that every hormone has to bind to a specific receptor. In order to exert that effect on the cell, that cell has to have a receptor for that hormone. It's very specific. And then when we look at those hormones binding to those receptors, a very strong affinity. That means that they bind together. Not only do they fit exactly, they also hold on together very, very well. Now that works to get our hormone to bind to the receptor, but because they hold on so strongly, we're going to be able to talk about, and I will just briefly mention this later on, is that once hormones and the receptor bind together, they create this complex that actually has to get broken down by the cell. So they bind very strongly. So here you'll see the term affinity, which refers to the binding of the receptors to the hormones to the receptors, and saturation, where hormones will come in. If you have a cell that comes in and it has 3 receptors, hormones that come in, we get 3 hormones that get released, they will bind to that receptor and they will hold on. So the saturation of the receptors is also key. Because what happens when that hormone binds to the receptor? You've got all of them bound together. That cell can't receive any more of those hormones because they've been fully saturated. So how do I get more receptors to be built onto, I'm sorry, no, I was about to start answering my own question. How do I get this cell to start producing more receptors? Well, that's where we get upregulation that comes in. We get the DNA that comes in, we get amplification of the signal, or we get an enzyme that comes in to produce proteins. Or rather, the enzymes will come in and start activating things to start creating more receptors to accommodate the amount of hormone that's present. So this will have a profound effect on what we're talking about with type 2 diabetes, where once we saturate that cell, or saturate all of our cells with insulin, these receptors can no longer bind to, and so they stop working as effectively, hence what we call insulin resistance. And we'll learn specifically what insulin does on Thursday. So this is going to help compare one and the other. So we have lipid soluble versus water soluble, and here this wraps it all up for you. Direct, indirect, on its effect on cell communication itself. The direct effect of lipid soluble is that we see increased DNA expression. Water soluble, we see the increase of our intracellular cell signaling molecule. Next thing we're going to take a look at is the physiological processes. What exactly do these endocrine hormones do? These hormones come in and they play multiple roles. So here you can see at least 5 examples of what these endocrine cells are supposed to do, and it really depends on the type of hormone. You'll see the balance and composition of volume of bodily fluids. So all this is going to be related to the urinary system. How can we regulate the solutes? If we have excess salts, how can we get rid of excess salts? Also, related to the amount of fluids. If we have excess water, so if our blood plasma is in a hypotonic state, how do we get rid of excess? How do we get rid of that excess fluid? And that's all related to either increasing the amount of solute or getting rid of excess water through urination. Regulation of metabolism and energy production is all through our thyroids, through regulating our metabolic rate. The rate and timing of growth and development. Here we're going to be able to see multiple hormones, testosterone. We'll see growth hormone and where that's produced. Exerting emergency control during physical and mental stress. These are things like fight or flight. So here we see the adrenals creating our corticosteroids, catecholamines. And oversee reproductive mechanisms. Here we're going to be able to see the effect of testosterone, estrogen, LH, and FSH. More hormones that we'll see later on. Other things that you're going to also be able to see, things that where we can increase the amount of ATP production, cell growth, as I mentioned earlier. So we'll see how that takes place on our later endocrine organs. Again, not too much on our prostaglandins and leukotrienes. These, again, were studied fairly recently, at least within the past 10-15 years. Because they are produced, again, by cells that do not have endocrine functions. So these typically our prostaglandins are involved in pain, platelet aggregation, fever, inflammation. Forget the next sentence afterwards. Don't worry about that. I don't want to get too much into detail about that. And so here you see the effect of platelet aggregation. So what starts off with a cell, you get a cut on that specific. If you do get a cut in the capillaries surrounding a tissue, This is where we get platelets that come around and have a paracrine hormone function, meaning these areas start to clot together. And they aggregate a bunch of platelets to come in and seal that wound specifically. Another thing is where you see aspirin come into play. So aspirin comes in and inhibits that enzyme for specific prostaglandins. And that will end up mitigating the pain as well as the inflammatory response. So we reduce these in effect. But because aspirin comes in and inhibits the production of those prostaglandins, it also has an effect on platelet aggregation. So typically aspirin has an effect on blood clotting. So you see it having that effect. Anybody that typically has an issue with blood clotting, they say don't take aspirin because that affects the production of those. This example is where I talk about upregulation. So this is upregulation and downregulation. Upregulation is basically the production of something in the cell itself, whether it's the receptors, the mRNA. This is the direct effect, as I mentioned earlier. Upregulation of a gene means the production of that, transcribing that DNA into RNA and translating that into a protein. So that's where we see typically where a hormone can result in the production of something. What is that cell supposed to be producing more of? Downregulation is an example of desensitization. What happens when we expose a hormone, multiple hormones, to a cell with receptors? As I mentioned earlier, that hormone, when it binds to the receptor, creates this complex, this hormone receptor complex that has to get broken down by the cell itself. So downregulation is an example of bombarding those receptors with those hormones. Okay? And think about what happens with type 2 diabetes, which is typically characterized by high blood glucose. So that glucose comes in and creates, triggers the insulin response, which we'll learn on Thursday, which will result in a bunch of insulin coming in and targeting cells to store that glucose. So glucose is supposed to be going into the cell. But now you have insulin coming in and binding to those receptors. And those receptors are then locked in with that hormone. And then, in order to accommodate that insulin response, that cell needs to upregulate the production of receptors. So it needs to come in and make more receptors in order to accommodate that rise in insulin. So as those receptors get built up, insulin can bind. But what happens when we have excess glucose where insulin gets produced? Insulin comes in, binds to, and that cell cannot produce enough receptors. So that's exactly where we see insulin resistance. Is that we have an overproduction of insulin and the receptors insulin not being able to bind to the receptors because they're all saturated. So therefore we see a decrease in the effect of the receptors. Those receptors cannot bind to insulin anymore, and insulin cannot work to reduce the amount of glucose in the blood. So what happens as a result? We have elevated blood glucose. Now something that you'll be able to see, the antagonist, and I'll describe what antagonist means on the next slide over. The antagonist hormone for insulin is glucagon, and glucagon is responsible for raising blood sugar. So because there's an effect on, there's a direct effect on insulin no longer being able to bind to the receptors, glucagon works normally, so it will start to elevate blood sugar. Again, something that we'll talk about when we get to the pancreas. So I started leading into the conversation about insulin and glucagon having an antagonistic effect. Let's take a look at the three main types of interactions of multiple hormones. All I've been talking about so far, I've been talking about one hormone binding to the receptor. Let's see how these hormones play together. How do they play together? Do they work for each other, or do they work against each other? Of which the first two are permissive effect and synergistic effect, basically increase the response of the two. The antagonistic effect is one in which one does one thing and the antagonistic hormone does the exact opposite. So the permissive effect means that the second hormone permits the action of the first. So taking a look at what I'm going to be talking about is epinephrine on lipolysis. This is the breakdown of fats. So here you can see epinephrine on its own cannot break down fat all by itself. But this is where T3 and T4 come in. T3 and T4 that are produced by the thyroids come in and activate epinephrine. So these two, when together, will allow lipolysis to take place. And those fats that get broken down into fatty acids. And those fatty acids will enter the Krebs cycle to produce, eventually down the line, ATP. Now, synergistic effect means that each of the hormones produce a response. Here we can take a look at norepinephrine creating a sympathetic response, epinephrine creating a sympathetic response, but when you put them together, they create a much more exaggerated response. It's not an additive effect, it's actually a multiplicative effect, meaning if you have two of these, it has 3 or 4 times the amount of an effect. So norepinephrine and epinephrine, both together, exaggerate that sympathetic response. Antagonistic effect, like I said, hormones that have an exact opposite effect, where insulin promotes glycogen formation or decreases the amount of blood sugar. Glucagon stimulates glycogen breakdown or increases blood sugar. So with that, let's take a look at how we can control hormones. So now we've talked about the production of hormones. We know how hormones affect their target cells based on whether they're lipid soluble or water soluble. We know that that's going to result in upregulation, the production of some sort of cellular component. We know what other cellular functions what other endocrine functions take place depending on what tissues are targeted. Now we can take a look at how we can control the amount of hormones. If we see the production of testosterone or we see the production of epinephrine, how do we control how much is released? And all of this is going to involve our feedback mechanisms, our negative feedback as well as our positive feedback, depending on what we're looking at. So looking at hormones that are produced by those endocrine tissues are released in bursts. So that way they get released, they get spurted into the circulatory system, and then they can make their way through the circulatory system. And you'll see the different mechanisms in place. Our signals from the nervous system. These are going to be our nervous pathways that lead into our first target organs. You'll see any chemoreceptors that will detect any changes that are taking place in the blood, or other hormones that will result. So hormone release by one tissue will trigger the release of another hormone. And that example we'll be able to see hypothalamus to the pituitary, pituitary to the other target peripheral endocrine organs. So this is all going to help trigger the release of what we're trying to produce. Negative feedback control is where we get the turning off of the production after the hormone levels increase. So increasing testosterone, we'll see how we can turn off the production of that back from where we started. Hence negative feedback, to keep us within a narrow window of a hormone. Positive feedback, on the other hand, is in which the increase of a hormone results in even more production of that hormone. Again, the difference between these two is the efferent pathway. Negative feedback, as you can see on here, reverses the stimulus. So here you can see blood glucose. So in this case, insulin responds to increased glucose. So let's say you just ate something. You ate something sugary, like a donut this morning. That donut makes its way into the gastrointestinal system. Those sugars get broken down into glucose, and that glucose makes its way into the blood. Now you've got a bunch of blood sugar, or sugar in the blood, and that's where insulin comes in. It's secreted by the pancreas in order to get insulin, or in order to get glucose to make its way into the muscle cell so that it can get stored as glycogen, whether it's the muscle or the liver. So we are extracting glucose from the surrounding interstitial fluid, specifically in the circulatory system, and storing it in muscle and liver cells. At the same time, insulin comes in and targets the production of glucose. So we effectively stop the liver from gluconeogenesis. So we turn that off, and we also turn off glycogenolysis. We want to turn off the enzymes that are involved in that. So insulin, if it's supposed to be storing insulin and turning that into glucose, so here we're turning it into, we're calling it glycogenesis. We want to store glucose and generate glycogen from it. We also want to turn off the enzymes that are involved in these processes. So what happens when glucose levels start to normalize? So once we pull out that blood sugar, we want to maintain a certain amount of glucose in the blood. So therefore, we're going to be able to see an antagonist hormone called glucagon, where glucagon can come in and start to raise blood sugar to bring it back to normal. So you'll see on the next slide over where we can maintain this value. And this value of what we're looking at in normal levels of glucose is 70 to 120 milligrams of glucose per deciliter of blood. That is that homeostatic value that you expect to see normal levels of glucose. Anytime your sugar levels drop, that's where glucagon comes in to raise blood sugar. Anytime glucose levels raise above that window, this is where insulin comes in and becomes active in order to reduce the amount of glucose. So you see how all of these come into play to regulate those levels right here. So this image you'll be able to see on a later slide what the stimulus is. We have chemoreceptors that will let us know how much glucose is in the blood, and whether if blood glucose is high, pancreas comes in, results in insulin being released, and that converts glucose back into glycogen to reduce the amount of glucose. If we reduce the amount of glucose too much, this is where glucagon, again secreted by the pancreas, comes in and reverses that process where we get glycogen turning into glucose to bring glucose levels back to normal. So you see this sort of teeter-totter balance of the two in order to maintain that right about 90 milligrams per deciliter of blood. Now the positive feedback system is this example that I used earlier, so I won't really go into too much detail about this because we've talked about this already. The stretching of the cervix during birth, we do have stretch-sensitive nerve cells in the cervix, and that we know by now. There are the muscle spindles involved. Muscle spindles are sending back to the brain, the central nervous system, and which is going to result in the production of oxytocin. And oxytocin, as a hormone, will have an effect on the uterine walls, which will cause contraction and pressing that fetus onto the cervix, which will result in more stretching. And that more stretching will come in and create an exaggerated response. So we have very positive feedback in this response that will trigger more oxytocin release. In the example that I mentioned earlier, this is a form of negative feedback, where increasing levels of glucose will result in the decrease of glucose levels. We're going to be producing these hormones. And so we're going to start off by looking at our endocrine glands. And these are specifically our pituitary, our thyroid, parathyroid, adrenals, and pineal glands. These are organs that are specifically geared for the production of hormones. That's pretty much all they do is just produce hormones. But if we take a look at other of these organs, these are the organs that have secretory cells. They, like the hypothalamus, the thymus, the pancreas, they have other functions other than just the production of hormones. Pancreas, will know, is for the production of insulin and glucagon, which we'll see later on, but is also going to be heavily involved in the digestive system. So they will be, specifically the pancreas is going to have exocrine as well as endocrine functionality. But what we're going to be looking at really is the central, some of the central organs. We'll see our hypothalamus. We'll take a look at the pituitary gland. As I mentioned, hypothalamus to the pituitary is going to be the starting of the system. We'll first start off with the hypothalamus, and when we look at many of the tracks later on, we'll see hypothalamus to the pituitary. So you'll be able to see the hormones that are involved being released by the hypothalamus that will signal the pituitary to create more hormones. So this is part of the hormone signaling that'll take place. And that pituitary will target either the tissues they're meant to target with a couple of the hormones that we'll be looking at, but a lot of the other peripheral endocrine organs, whether we're talking about the pancreas, ovaries, testes, kidneys, you name it, small intestine, skin, heart, all of those will be released by the pituitary itself. So you'll see how we can see that signaling that leaves the pituitary. So the hypothalamus, as you know, is the major area that's going to govern homeostasis. It's got numerous nuclei that are involved in the regulation of many systems that we're going to be looking at. So it's going to receive lots of sensory information from many areas. You can see the thalamus, which we mentioned, is a sensory relay area. RAS being the reticular activating system, which is all related to awareness, as well limbic system, which is tied into emotion. So all these tracks that lead into specifically the hypothalamus. You see a lot of innervations that lead to that area. Okay. So what we'll take a look at is we will start off with the hypothalamus. The hypothalamus we know is going to be consisting of neurons that are going to travel to the next target, which is the pituitary gland. So the hypothalamus controls the pituitary gland, and the pituitary gland is right below the hypothalamus. So thalamus, you'll see underneath the thalamus, we have the hypo meaning low, so below the hypothalamus, and then below the hypothalamus is going to be the pituitary. The pituitary is also known as the hypophysis. So while we'll do a quick anatomical breakdown of the pituitary as it's broken down to its two different parts. We see the anterior portion called the anterior pituitary. And that anterior pituitary, here you can see the front portion of the pituitary. So this is the front part of the brain. This is the anterior pituitary. The back portion is called the posterior pituitary. I'll talk about the terms on here called adenohypophysis and neurohypophysis. So the term hypophysis really just relates to the term pituitary, while the other, adeno and neuro, are going to represent the cellular makeup of the cells in that particular tissue. And you'll see that they differ quite a bit. The anterior pituitary is actually connected to the hypothalamus. They're actually, if you were to take a look at the hypothalamus, which consists of neurons, you'll see that the anterior pituitary is also consistent and connected through this hypophyseal portal system. So there is a connection between the two. And so in the anterior pituitary, you'll see the anterior pituitary is actually a different makeup. It actually consists of endocrine cells that secrete hormones into the capillary bed surrounding. So there is no actual real direct anatomical connection between the hypothalamus and the anterior pituitary. And they're going to rely on this portal system, which we'll describe later on. The portal system is what connects two capillary beds together. We have a capillary bed that surrounds the hypothalamus. They converge into the portal system, which are basically portal veins that connect the next capillary bed. So in this case, because our hypothalamus consists of neurons, and our anterior pituitary consists of endocrine, the way they communicate is through this hypophyseal portal system. So let's take a look at how that works. So in the hypothalamus, we have, here we can have this hypothalamic neurosecretory cell. These are neurons that are meant to secrete hormones. And they don't, they're kind of modified neurotransmitters that get dumped into the capillary bed surrounding the hypothalamus. Those hormones that are released are called releasing hormones. So that's the way they communicate. Now, looking at the anterior pituitary, it's receiving those hormones, those releasing hormones from the hypothalamus. And those releasing hormones are going to come in and target 5 types of cells. And those types of cells that we're going to be able to see in the anterior pituitary, if we literally chop up the anterior pituitary, you can see different regions are going to contain these 5 different types of cells. And they're going to create 7 types of hormones, of which you can see on the very bottom. 1, 2, 3, 4, 5, 6, 7. And we have 1, 2, 3, 4, 5 types of cells. And based on what they target is going to be the naming convention for those types of cells. You'll see these cells creating tropic hormones because they target other endocrine organs. So releasing hormones that will be released by, I'm just going to call them RH, produced by the hypothalamus, will come in and target each one of these 5 trophic cells, okay, of which you're going to see somatotroph, thyrotroph, gonadotroph, lactotroph, and corticotroph. They'll come in, and those releasing hormones will come in and trigger them to come in and produce tropic hormones that will come in and target other endocrine cells. So what are those names of those hormones? We'll be able to see. Human growth hormone, TSH, which is thyroid stimulating hormone, FSH, LH, prolactin, ACTH, and MSH. I'll be describing that for you on the next table over. Basically, all together, this is all one table in which the left column represents the hormone, the releasing hormone that's produced by the hypothalamus. You'll see the hormone that's released by the adenohypophysis, which is the anterior pituitary. So here you see that hormone coming in, triggering those trophic cells. That will release another hormone that will make its way over to the next target. And that target can either be another endocrine, or target a tissue specifically. So here you can see, looking at the names of the hormones of the hypothalamus, let's start off with the left column. The hypothalamus hormones are really easy to remember because they're either releasing hormones or something that we'll talk about are going to be inhibiting hormones. Either they releasing hormones that release hormones here, or they release inhibiting hormones that inhibit hormones being produced by the anterior pituitary. So the ones I'm going to come in and point out, I'm just going to highlight these for now. I'm going to highlight the human growth hormone. I'm going to highlight prolactin, and I'm going to highlight MSH, which is our melanocyte stimulating hormone. And I'll talk about why I'm highlighting this in just a little bit. So here you can follow the pathway. Growth hormone releasing hormone is produced by the hypothalamus to trigger the production of human growth hormone within the anterior pituitary. Which cells are producing human growth hormone? that would be our somatotropes. So they produce that tropic hormone called human growth hormone. It's also called somatotropin. Somatotropin meaning, somato meaning body, soma. Tropin because it's a trophic hormone. And what that's going to do, it's going to stimulate the secretion of insulin-like growth factors. And if you ever study growth or any of these hormones, IGFs are something that's of interest. They're really interested in this because it's heavily promoting growth. The next one, thyrotropin-releasing hormone, is going to come in and stimulate our thyrotropes. The cells, again, in the anterior pituitary, which is going to create a hormone called thyroid-stimulating hormone. And if I were to ask you the thyroid-stimulating hormone, what is it going to stimulate? The thyroids. So it comes in and stimulates the thyroids to produce hormones that are produced by the thyroid gland, which we'll talk about later, which is T3, T4. Prolactin-releasing hormone is going to produce, again, prolactin-releasing hormone releasing prolactin from our lactotropes. And prolactin is going to stimulate breast growth. So, looking at each of these, gonadotropic releasing hormones, both producing follicle-stimulating hormone and luteinizing hormone from the anterior pituitary. Again, this is going to be involved in reproduction, not only development of gonads, but as well as our sperm and eggs. And the last one, corticotropin-releasing hormone, is going to come in and stimulate our corticotropes in the anterior pituitary, which is going to result in the production of adrenocorticotropic hormone. And what is that supposed to stimulate? The adrenal cortex. So that's the next target over, is that ACTH is going to target the adrenal cortex. And here we'll be able to see 3 different types of corticoids. Because we're involving the cortex, you'll see the hormones that are produced are called corticoids. And we have mineral corticoids, glucocorticoids, and androgens, depending on which layer of the adrenal cortex we're looking at. The last one we're going to be looking at is corticotropin-releasing hormone. And again, corticotropin-releasing hormone is going to stimulate our corticotropes in the anterior pituitary. And that will result in the production of what's called melanocyte-stimulating hormone. And what is that going to stimulate? It's going to stimulate melanocytes. Where do we find melanocytes? Melanocytes are involved in the skin. So it's all for the production of pigmentation. So each of these, the ones that I did not highlight, are all going to come in and stimulate another peripheral endocrine organ. Thyrotropin-releasing hormone comes in and stimulates our thyrotropes in our anterior pituitary, which is going to stimulate our thyroids. Our follicle-stimulating hormone is going to come in and target the testes or the ovaries, depending on male or female. Ovaries and testes. Corticotropin-releasing hormone is going to target the adrenal cortex. Now take a look at the ones that I highlighted in yellow. We have the skin. We have the breast, mammary glands. And for human growth hormone, we're targeting things like bone, muscle. Those typically are not endocrine glands. So what we're going to have to take a look at, as I mentioned earlier, in each of these systems, we're going to have an inhibiting hormone that comes in. So in this case, we have a thyroid. We're going to see the production, again, something we'll talk about later. Bless you. T3 and T4. T3 and T4 are going to be the products, are hormones that are produced by the thyroid gland. When we have T3 and T4 present in our blood, we got to have something that's going to come in and inhibit the hypothalamus to limit the production of T3, T4 through the form of negative feedback. In the case over here, the production of testosterone. Testosterone is going to come back and turn off the hypothalamus from producing our gonadotropic releasing hormone. So in these cases, you're going to be able to see, and all of these are going to be inhibiting hormones that come back. They'll come back and inhibit the hypothalamus. The reason I highlighted human growth hormone, prolactin, and melanocyte stimulating hormone is that these are not endocrine cells. So we're going to be able to take a look at what is going to inhibit the growth hormone releasing hormone. So we're going to be able to see something called GH inhibiting hormone. Here we're going to be able to see our prolactin inhibiting hormone that comes in to turn off the production of those hormones later on. Okay. So that should make a little bit sense when we get to it. We look at thyrotropin releasing hormone. We're going to see thyrotropin inhibiting hormone to inhibit the production of that. It's either a releasing hormone to stimulate or an inhibiting hormone to turn off. So let's take a look at our first one. Is human growth hormone. Human growth hormone is a hormone which, obviously, by just looking at the name, you know it's involved in growth. It not only is involved in growth, it also takes a heavy role in metabolism. Human growth hormone is something that actually has hit, especially in the bodybuilding world. It's something that has been sought after, especially after testosterone. We knew that exogenous testosterone, injections of testosterone, is involved in heavy growth. Best case example, Sylvester Stallone. How that guy was able to remain big, despite being in his, I think, 60s now, 60s, 70s, being as old as he is, has taken human growth hormone injections in order to increase the growth of skeletal muscle mass. So it's heavily involved in that. And if you take a look at what it does, it's pretty self-explanatory. Growth of skeletal muscle, growth of bone, and then in order to contribute to the growth of skeletal muscle mass, we obviously know that these are proteins, and so proteins will need to undergo amino acid uptake. So the cells need to take in these amino acids to start producing those proteins. Just like that, just like the transcription and translation that we were talking about at the end of our cellular physiology lecture. This is how we create proteins. That is pretty self-explanatory, but it does have an effect on metabolism as well. So what human growth hormone does in terms of our metabolism is it stimulates gluconeogenesis. Okay. So this allows the release of glucose. So we're now increasing the amount of glucose, and we're also increasing the amounts of fats, fatty acids that can be used for energy. So looking at these two, these are involved as metabolic substrates to produce ATP. So it's heavily involved in metabolism. Why doesn't it use protein? Well, it's not using protein because protein is going to be involved in the production of skeletal muscle. So the stimulation for this in these cases typically are low blood sugar. Low blood sugar will stimulate the release of human growth hormone. Sympathetic activity. Any heavy exercise of any kind or any stressful situation initially will result in heavy sympathetic activity, fight or flight, that will result in lipolysis and also gluconeogenesis to produce our metabolic substrates to churn out a bunch of ATP. But at the same time, it's going to be protein sparing because that's going to be reserved for the other function for growth. Typically is why, especially when looking at exercise programs, you probably have seen heavier intense exercise, what they call high intensity interval training. These are things where you have like heavy bouts of sprinting mixed with walking, and it's just intervals back and forth. Why is that so important? Well, because those result in the highest sympathetic activity. There's a lot of fight or flight that comes in with it, and it also results in low blood sugar, which also stimulates the amount of human growth hormone. The other thing that's also important with human growth hormone is deep sleep. Hence why getting at least where we saw deep sleep take place, most of it you saw within the first 4 hours. And also another reason why you see children sleep so much. For any of you that have children, or at least all of us at some point have been children, we know that during your growth spurts, you have a tendency to sleep a lot more. Why is that important? Well, that's where growth hormone comes in. The production of growth hormone involved in which is mostly prominent at times of those growth spurts and puberty, is where we're going to see the biggest rise in the growth of skeletal muscle and bone. Okay? So we need human growth hormone specifically for that. Also something that I've read as well, something that actually limits the amount of human growth hormone, drinking. Actually also plays a big role in that. I remember looking up studies on that talk about heavy nights of drinking will actually limit the amount of human growth hormone. Human growth hormone and growth. Let's take a look at how this plays a role in bone growth. And here you can see in the growth of long bone growth. So this is related to the lengthening of the bone itself. There are specific areas on the bone which are called the epiphyseal plates. These plates right here are areas that are a combination of cartilaginous tissue that's supposed to fuse and turn into hard bone itself. So these epiphyseal plates is what we call, and you don't need to know the term, but it's called endochondral ossification. So this is where we get the cartilage that starts to harden, and as this starts to harden, it's going to create more plates, and it creates this whole elongation of the bone itself. Now that is specifically how bone growth takes place. Again, the lengthening of the bone. Let's start to take a look at what happens with excess growth hormone. And this is on my next slide over, but I'm going to draw it on my slide prior. What happens with excess growth hormone? Well, taking a look at what it's supposed to do in the first place. Excess growth hormone will result in excess glucose. So typically, people that have too much growth hormone will have mimic diabetes, meaning high blood sugar. So typically results in blood glucose. And because we got a lot of excess glucose, you're also going to see a lot of insulin being produced by the pancreas. So the pancreatic cells that are going to be involved, which we'll learn later on, are called beta cells of the pancreas, are going to continually produce insulin as a result of excess blood glucose due to excess human growth hormone. So this term that they typically refer to people that have excess growth hormone, whether it's endogenous growth hormone, overproduction of growth hormone, or intake of exogenous growth hormone. People that take injections of growth hormone will have what's called beta cell burnout. And it mimics that of diabetes. Let's take a look at the effect not just on metabolism, let's take a look at what it does for growth. So the first term you're going to see on here is acromegaly, and megaly means big. So what happens with excess human growth hormone is just over-enlargement and over-elongation of the bones, specifically in the face, jaw, cheeks, and the hands. So all of those areas are continually growing, and the other areas that stop growing is long bone growth in the extremities. After a while, that stops because this area, after a certain age, completely ossifies. It turns to bone, and no longer will growth take place on the length of the bone. But you'll also start to see a lot more calcium deposition taking place on the sides of the bone itself. So typically with acromegaly is you see the thickening of the bones. And eventually what's going to happen is you see ossification of the appendages. So if you're to take a look at the fingers, the toes, eventually over time what's going to happen is going to see solidification of this. Anybody remember the guy in the top right? No? Andre the Giant. And it's just more and more I teach this class, I notice less people start to recognize this guy. This guy from the 80s. This is Andre the Giant. And Andre the Giant was a giant. He actually had acromegaly, but he had this overproduction of human growth hormone. And what happened with him was he had massive size, and as he started to grow older and older, you can see he started getting thicker, and you can see the prominent cheekbone starting to stick out quite a bit. And then you also see the ridge, the upper eye ridge that started to form that was very prominent. This other guy on the bottom right is Richard Kiel, Jaws from the James Bond films. I forgot which movie it was in. But you can also see the same sort of features, a very Neanderthalic look to their face. They have this prominent eye ridge bone, you can see the cheeks, and then the jaw, very strong jaw features. And all of that is due to that excess bone growth in that area. And unfortunately, what happens to people that have this, another side note about Andre the Giant. Andre the Giant was probably the biggest drinker ever. He used to drink like 30 beers in a setting and it wouldn't really affect him. He was the kind of guy that would apparently he used to go to bars and used to buy drinks for everybody, but he was the guy that had the biggest tab of them all because he took in so much. And he was a huge guy. But a lot of the reason of why he also drank a lot too, this guy was in a lot of pain. And there's a reason as time went on, he got older and older. It became very difficult for him to walk because areas of the knees, the toes, the feet, those areas that are supposed to have cartilage became ossified. So it became increasingly difficult for him to move because of the lack of flexibility he started to have. And you notice as he got older, he had to rely on a cane because he just couldn't move as much anymore.
What do you mean ossified? Like
what's that?
Thin?
Ossified meaning the turning into bone. So any areas, think of what's happening with the joints, right? They're supposed to look like this, and eventually more bone growth, and eventually those fingers couldn't really move as much anymore. And so there's all this bone growth on the outside and less flexibility. And with that becomes a decreased quality of life. It becomes increasingly difficult for him to get up in the morning, to move around. And of course, this guy is an actor, so he made most of his money through acting and wrestling. And so because of his limitations in movement, he started living a harder life towards the very end. And that's really it about him. Now, the opposite of that with human growth hormone imbalance is pituitary dwarfism. And they call it pituitary dwarfism because growth hormone is not produced as much. And so the pituitary does not produce, the somatotrophs do not produce as much human growth hormone. So you see a huge stunting of their growth. What you typically see is that, especially during childhood, you see very slow growth. So people that have pituitary dwarfism look like children. They just can't grow as fast. And I don't want to confuse this for any other form of dwarfism, but let's show you an example of this, the world's shortest girl who had I think one of the worst cases of pituitary dwarfism.
मुझे 18 साल की हूँ तो 18 साल की हो गई हूँ तो मुझे ऐसे अपने ऐसे लगता है कि मैं भी बड़ी हो गई हूँ. जब मैं बाहर जाती तो सारे लोग मेरे को ऐसे भीड़ जमा के खड़े हो जाते. इतनी सी लड़की इतना सब करती, इतने लंबे बाल, इतने बड़े नाखून, इतना मेकअप, सब पूछते रहते. इतना सब कैसे करने की बात करती क्या, चलती क्या, सब.
So that's really it. I can't see too much of that. So that's an imbalance. So that's the opposite case of it, where we saw Andre the Giant with excess growth hormone. You see the world's shortest girl with low growth hormone. Now that we know what growth hormone does specifically in terms of growth as well as metabolism, let's take a look at how we can regulate the amount of growth hormone. Again, we don't want too much of it, we don't want too little. So therefore, we're going to be able to see the mechanisms in place to regulate how much we can produce of it. So here you can see the image to the right, which is going to graphically show you how we, to the left, which is going to result in the increased amount of growth hormone, and the right side in red is going to show you the inhibition of the production of growth hormone by the anterior pituitary. So what triggers do we start off with? Again, we have to start off with our sensory modality. Here we can take a look at our receptors. or the sensory or afferent pathway that leads to our control center, which is going to be our hypothalamus. So these chemoreceptors are going to detect low blood sugar. And what starts off with low blood sugar was going to result in the production of our growth hormone releasing hormone by the hypothalamus. That growth hormone releasing hormone is going to get secreted into the capillary bed in the hypothalamus, and it's going to make its way into the portal system back into the capillary bed surrounding the anterior pituitary. So that growth hormone releasing hormone will come in and target the somatotropes. The somatotropes then are going to release human growth hormone, which is now going to raise blood sugar back up. So here you see what starts off with low blood sugar results in increase in blood sugar. Okay? So here that efferent pathway is the reverse of what the stimulus is. So this is a form of negative feedback. So blood sugar raises back up. And what happens in the cases where we get, when we start to raise blood sugar above normal? So here we have hyperglycemia. So where we referred to before was hypoglycemia, low blood sugar. Hyperglycemia is the increase in blood sugar. So the increase in blood sugar is going to result in the production of growth hormone inhibiting hormone. And that growth hormone inhibiting hormone is not just produced by the hypothalamus. There's very little amount produced by the hypothalamus. It's called somatostatin. It's a name for growth hormone inhibiting hormone. Where we're going to be able to see somatostatin later on, we're going to be able to see this in our gastrointestinal tract. We'll be able to see this specifically in the small intestine. It's one of the hormones that's produced by it. A little bit of it by the hypothalamus, but mostly from the small intestine. So somatostatin comes in and inhibits the production of growth hormone by those somatotropes in the anterior pituitary. So now we get a lower level of human growth hormone. Low levels of growth hormone will cause blood sugar going back to normal levels. So high blood sugar results in decreasing blood sugar levels and therefore bringing us back to that window. Again, 70 to 120 milligrams per deciliter. We want to keep that around there. This is one part of blood sugar regulation that's involved with growth hormone. We'll be able to see how this takes place with insulin and glucagon later on. Now, why did I highlight this earlier? I want to go back to human growth hormone. Human growth hormone, in this point right here, the end product of human growth hormone in relation to metabolism is all related to sugar, the regulation of sugar. Sugar can't go back to the hypothalamus to inhibit at any point, so that's where we're going to have involving that growth hormone inhibiting hormone. That's why I drew that out, why I highlighted specifically this right here, where growth hormone inhibiting hormone, which we'll learn later on as somatostatin, comes into involve negative feedback to stop the production of growth hormone. Now prolactin, the second of our hormones, prolactin, again, another anterior pituitary hormone that has its target on mammary glands. So again, produced by the anterior pituitary, and its target are the mammary glands. So prolactin is specifically involved in, if you take a look at the term lactin, we know is related to the production of milk. Two things involved in lactation. One, milk production. The second, called milk letdown. Now, milk letdown is the release of milk. Prolactin is all involved in the production of milk. So here, with prolactin, you would see the increasing size of the mammary glands. As they swell up, you'll see that they're involved in the production of milk. So this is where PRL takes its effect. This one, milk letdown, we'll learn as produced by the posterior pituitary, is going to be involving oxytocin. So two hormones involved in lactation. So here we would typically see prolactin levels. They'll fluctuate quite a bit in women. That varies based on menstrual cycles. So at certain stage of the cycle, typically women will start to mention that their mammary glands will start to expand quite a bit. And then that's all related to fluctuating levels of prolactin. Now, what we also notice about pregnancy is that towards the latter portion of pregnancy, you start to see rising levels of prolactin. And that, again, is typically something that you would see that's involved in preparation for milk production that takes place after pregnancy. Because there's so much involved in prolactin, it's not uncommon to find those same prolactin levels. Since we start to see very large levels of prolactin, you start to see neonates, babies that are freshly born, with also high levels of prolactin. You also notice that those babies have tendency to also have increased mammary sizes as well. So it's not uncommon for newborns to have profound mammary glands that start to decrease over time because their prolactin levels go back to normal. Sometimes these neonates can start to produce what's called witch's milk. It's a little bit of milk that's actually produced by the neonates due to that excess prolactin. Now, what's going to regulate the amount of prolactin is prolactin inhibiting hormone, PIH, because milk doesn't come back and turn off the production of prolactin in the anterior pituitary. What needs to be involved is another inhibiting hormone called PIH. And what that is, and what we've learned later on, is that prolactin inhibiting hormone is dopamine. So dopamine comes back in, and we'll learn that dopamine comes back to inhibit the lactotropes, again, in the anterior pituitary to reduce the production of prolactin. So this is, again, our form of negative feedback to limit the amount of prolactin. And again, something, the menstrual cycle, pregnancy, as well as post-orgasm. Something I want to mention during menstruation is the decrease. So typically we would see in menstruation, you see a decrease in prolactin inhibiting hormones. So what we learned later on to be dopamine. And we know dopamine starts to affect specific areas in the reward center. It's one of those neurotransmitters that's also heavily involved too. So typically with the decrease in dopamine, we know that dopamine, especially in psychology studies, have noticed that dopamine is a reward motivating behavior seeking hormone. Especially people that with addiction will look at finding things that will increase levels of dopamine. Because we see the decrease of levels in dopamine, you typically would see symptoms of shaking, muscle stiffness, all things that come in and stimulate the substantia nigra. Not only does that also during menstruation, when we start to see increasing levels of PIH, you start to see behaviors that are typically behavior seeking, things that promote reward. Something that I've tied into this is why women sometimes have a certain preference for certain foods during menstruation. And something I've seen before is chocolates that can also come in and stimulate the reward as well. Again, tied into dopamine release. The other anterior pituitary hormone that we're going to talk about is melanocyte stimulating hormone, MSH. And these, as you mentioned earlier, are going to stimulate the melanocytes. The melanocytes that are present in our skin as well as our hair. So these are, as I easily mentioned, is just involved in our pigmentation. But where we start to see increased MSH is through pregnancy. So typically women when are pregnant are going to have a form of hyperpigmentation.
That's why they say they have the glow or something like that?
The what?
The glow of pregnancy.
Glow is not more related to more blood flow to certain areas. This really is more about pigmentation actually in the skin itself. So another thing why also pregnant women will notice that they have awesome hair. Like their hair becomes thicker and much more vibrant, and it's due to that pigmentation in there. Another thing that you also see in pregnant women is an area I think they refer to as linea nigra. It's this whole area right here that forms in pregnancy. Again, just a line of pigmentation that typically forms. I believe that was called linea nigra. And all this hyperpigmentation that happens in pregnant women is called cloasma gravidarum. Again, these terms you really don't need to know. Just Other areas that MSH can also come in and stimulate. While it does mostly stimulate the melanocytes, it's also tied in a little bit to appetite and also tied in a little bit to sexual arousal. But those again are very minor. Okay. So now that we've talked about the entire anterior pituitary, again looking at MSH, prolactin, PRL, we looked at growth hormone. Let's take a look at the neural hypophysis. The neural hypophysis being the posterior pituitary. So looking at the posterior pituitary, we're going to take a look at the hypothalamus and the posterior pituitary. The reason it's called the neurohypophysis, hypophysis referring to the pituitary, is that the posterior pituitary is actually similar in cellular and tissue structure with that of the hypothalamus. While the anterior pituitary is comprised of endocrine cells, our posterior pituitary is comprised of neurons. So here you would be able to take a slice. You would see that these would mostly be consisting of neurons, especially axons projecting from the hypothalamus. And I won't necessarily have you guys remember any of the nuclei that are present in the hypothalamus, paraventricular, neurosecretory, and the supraoptic nuclei. Not important, but we know that the cell bodies that are located in the hypothalamus project directly into the posterior pituitary. So here you can see it's releasing the hormones that originate from the hypothalamus and release them into the posterior pituitary out into the surrounding circulatory system. The two main hormones that are released are oxytocin and ADH. So here you see they are released by the axon terminals located in the posterior pituitary. Let's take a look at the two different hormones, oxytocin and ADH. So oxytocin is that second portion of milk production by PRL, or lactate releasing hormone, and then milk letdown is involved with oxytocin. Where this targets specifically is the uterus and the breast. Those are the two main areas for oxytocin. So for the uterus, as I mentioned earlier, for positive feedback, stimulating uterine contractions. So oxytocin typically is something that you would notice that comes in that, as I mentioned earlier, results in stretching of the cervical wall. It results in oxytocin being released, and then oxytocin comes in and targets the uterine wall to cause contraction. And that contraction again presses against the cervix, which results in more oxytocin being released. So typically what you would see, especially towards approaching birth, is that we have increasing levels of oxytocin. And what oxytocin does, again, causes more and more contractions. So something that they typically would use in order to stimulate childbirth is a similar hormone called Pitocin. Injections of Pitocin would result in uterine contraction. So if they want to induce labor on somebody, Pitocin is the drug that they use. Another thing about oxytocin is that it's involved in human bonding. If you ever hear about what you can do to stimulate oxytocin levels, is skin contact. Skin-to-skin contact, not with yourself, but specifically with others. Now, oxytocin release is something why when newborns are born, they typically will place on the mother's chest. And that initial contact starts to result in excess oxytocin production, right? So that oxytocin release not only starts it from a psychological perspective, increases that mother-child bonding, but it also releases two functions. Number one, to release milk, the milk letdown that actually takes place. So that's a reason why they have mother and child together right after birth. The next thing is it also continually stimulates uterine contractions in order for ejection of the placenta itself. So that's what contributes to that portion. So all that results in the stimulation of milk letdown, which results in breastfeeding, and breastfeeding again results in more oxytocin. Something that also ties into orgasm. Something that oxytocin is also released due to skin-to-skin contact results in the set point of where orgasm takes place. I won't really go into too much detail about that, but there is other weird phenomenon that I remember reading about when I was putting these slides together, is that it's not uncommon also for women during milk letdown or release, that it can actually also stimulate orgasm just due to the levels of oxytocin circulating through their blood. I didn't really want to go too much more into that because then that started leading me down the dark path of the internet where I'm like, I don't know if I want to be reading this stuff because that's stuff on my internet search history that I
It
want
was
to
on
wipe
Grey's
out.
Anatomy last episode, and it also helps at childbirth.
Yeah, that's exactly it. That's what oxytocin is for. And then the all the other alternative that they can inject, the other analog version would be called Pitocin. The last of these is that we'll talk about is antidiuretic hormone. And antidiuretic hormone, if you look at the name, it's pretty easy to determine what its function is. We know that a diuretic results in the release of water through urination and through sweat. An antidiuretic hormone works opposite of that. So what it does, and we'll learn in our urinary lecture, is that ADH, antidiuretic hormone, comes in and binds to the receptors in the collecting ducts. These are the areas that are going to be taking water back into the circulatory system. So what that's going to do, it's going to come in and target it. It's also going to target the sweat glands to minimize the amount of water that we lose through sweating and urination. So an antidiuretic is to conserve water. So it's all related to how we conserve water. We'll learn about another hormone later called aldosterone, which is going to be related to not only conserving water, but also conserving salts. So this antidiuretic hormone is purely for the regulation of water levels in our body. ADH also has a secondary function involved in the constriction of the arteries. So antidiuretic hormone is also something that you would see in cases where we see blood pressure loss. We see rising levels of ADH. So that if we lose blood, let's say in the case where we're losing a lot of blood, right? We want to minimize the amount of water loss, so therefore we're going to employ an antidiuretic hormone. Combine that with arterial constriction, will now start to raise blood pressure back up in the cases where you're losing blood. So the other term for antidiuretic hormone that you might see in the book, it's also referred to as vasopressin. Vaso being vessel, pressin related to pressure. So these things are going to be things that are employed in order to maintain blood pressure. How do we regulate the amount of ADH is all related to the tonicity of the blood. Again, why we talked about our hypotonic and hypertonic. So hypertonicity, where we have high amounts of solute, right? If we have high solute, low solvent, or in this case water, what do we do in order to raise us back to isotonicity? Well, if we already have high solute, all we need to do is raise the amount of water, right? So raise the levels of water, And that should bring us back to normal tonicity. We may have high solute, but raise our water, we create it's water retention. Retain water, and that should help bring us back to isotonicity. So how do we increase our water retention? That's where ADH comes in. So the secretion of ADH by the posterior pituitary results in less water loss through the kidneys, less water loss through our glands, and also drives arterial compression. And that compression comes in to raise blood pressure. Now the reverse of this, low solute and high H2O, in a hypotonic situation, all we would need to do, if we can't regulate the amount of solutes, we can decrease the amount of H2O. So what we want to do with that is decrease the amount of H2O. Therefore, the only way to get rid of excess H2O is to decrease the amount of ADH. And that decrease in ADH, so we limit the amount of ADH, that results in diuresis, the excess water loss through the kidneys, as well as sweat. So all these play a role in the regulation of that, and you can see that in our graph to the right. What receptors are feeding into the hypothalamus are chemoreceptors, specific chemoreceptors called osmoreceptors that detect the levels of tonicity, the amount of solute in respect to the amount of water in our system in order to regulate them. You think about these also in the cases of high solute, low H2O. You would typically see this again in cases where you see excess salt intake. Excess salt intake, you've had a very salty meal. Typically what that ends up doing, you end up retaining more water. You get bloated, you feel more bloated. And that's again due to increasing levels of ADH. In areas where you just drank way too much water, right? So you have low solute, or let's just say regular solute, but a higher amount of solvent. If you just drank way too much water, what that's going to do is going to limit the amount of ADH production, right? In order to eliminate water through sweat as well as the kidneys. And again, our osmoreceptors come in and feed that back into the hypothalamus to regulate the amount of ADH production in the pituitary that will target tissues to do what it's supposed to do, is to regulate the water levels. So all of that we've talked about is pretty much all of our central endocrine glands. Again, talking about the hypothalamus, as well as the posterior pituitary and anterior pituitary. Again, when we meet on Thursday, we'll talk about all the other tissues involved. Thank you for your guys' attention.